Okay, thank you. As everyone else has said, it's really a pleasure and honor to be here, and thanks to the organizers for pulling together a very interesting cross-cultural meeting. So the title of my talk says, Embryos, Embryoids, Chimeras, Gene Editing, and their promise for health and ethical concerns, all in 30 minutes or less. Well, let's see what we can do. I want to set this in the context of the embryo. So I'm going to, many of these things you've already heard about, but I want to remind you that about this stage of development, the blastocyst that we've heard mentioned today. So the mouse, this is a mammalian blastocyst. This is actually a mouse blastocyst. It consists of 3 cell types. And what we know in the mouse, and it's true in the human as well, is that those 3 cell types, when this embryo has about 100 cells, has not implanted in the uterus, these 3 cell types give rise to all the cells of the later conceptus. And the outer cells give rise to the placenta. These are the trophoblast cells. This green cell layer is a primitive endodome, gives rise to part of the yolk sac, and it's only these little pink cells here that are the pluripotent cells of the embryo. And my lab and many others showed many years ago that these indeed are distinct lineages by this point. These epiblast cells are pluripotent, and in chimeras make the entire fetus. So it's that pluripotent capacity of the epiblast that, of course, has been what has led to embryonic stem cells, first in the mouse, and then, most famously, by Jamie Thompson in the human, whereby taking human blastocysts, he was able to get out those epiblast cells and transform them in culture into permanent pluripotent stem cell lines, dividing indefinitely in this undifferentiated epiblast-like state, but of course being able to differentiate into many, many different cell types of the body, in the same way that in the mouse we were able to show that ES cells could actually make an entire mouse. You can't do that with human, but their pluripotent capacity has been what's driven the whole pluripotent stem cell field in the years since Jamie's first discovery. So human ES cells, as we've heard, the work was controversial. Not controversial in the sense that it opened up incredible opportunities for regenerative medicine, but controversial because of the use of human embryos. So the concept that you would take a human blastocyst with human potential and turn it into a stem cell led to many controversies in many different jurisdictions. And of course, we know that mostly human embryonic stem cells are derived from IVF embryos that are no longer used for reproductive purposes and that would otherwise be discarded. Full informed consent, but again, as we've heard today, then and still now, there are legislative restrictions in many different jurisdictions about the derivation of human embryonic stem cells and indeed the use of such cells in culture and in regenerative medicine. So of course that all changed with iPS cells. Game changer number 1. Game changer of iPS cells from Yamanaka, showing first in the mouse and then in the human that you could take any adult cell essentially that still had a full nuclear DNA complement and turn back the clock. Turn back the clock by giving that cell expression of some of the genes that we knew were important for pluripotency in the embryo. So you go back to the blastocyst. Everything comes from the blastocyst. Those genes, those transcription factors, were able to reprogram adult cells and turn them into cells that in many, many different aspects resemble human embryonic stem cells. And as we've heard, they can be expanded indefinitely, and just like ES cells, can differentiate into many different cell types, with the long-term goal, potentially, of rejection-free transplantation, in that we could make iPS cells from everybody in the room, and you could have your own store of cells for future use. That's really probably never going to be a clinically applicable approach, too expensive and too slow. But iPS cells, as we've just heard very nicely, are wonderful systems to model human disease because you can access all the genetic variation in the human population and take it right through development from early stages to differentiation. Drug screens, many different aspects become applicable. So we haven't mentioned this at all, but we've talked about the power of regenerative medicine, and since 1998 there's been a lot of hype around pluripotent stem cells and how they really are going to transform our treatment of degenerative diseases and serious injuries. But is it true? Are we actually seeing now the fruits of all this investment? And I would argue that the answer is yes. Just now, really in the last few years, we're finally seeing that a number of different pluripotent stem cell-derived therapies are in clinical trial. And here are just some of the trials that are ongoing worldwide. Spinal cord injury, macular degeneration, Parkinson's, we've just heard about, diabetes, and even heart disease, replacing damaged heart muscles, not yet in human trials, but very promising preclinical work here too. And I just want to point out that in all of these trials, with the exception of some of those going on in Japan, actually use human embryonic stem cells. So pluripotent stem cells provide us with new frontiers. They can help treat degenerative diseases and model development and disease in the dish. But it is true that almost all of those trials are being done with human embryonic stem cells, so the ethical concerns around human ES cells still persist. So what else can we do with pluripotent stem cells? We heard a little bit about this morning. So let's think about taking human stem cells and not using them to study disease or treat disease, but what else can we use them for? Well, interspecies chimeras have come to the fore. The concept that since these cells are pluripotent, could we get them to derive tissues and organs in another animal such that you could develop in a sort of xenograft a situation where you could generate a human organ that could be used to transplant back into a human. And this work really has come from what has been done in mouse and rats for many years. In fact, when I was a graduate student, my supervisor made the first mouse-rat chimeras. But more recently, that work has been extended by taking mouse embryos and injecting a mouse blastocyst with rat embryonic stem cells, and showing that you can generate a mixed-up animal. The pluripotent cells mix up with the pluripotent cells of the blastocyst and essentially give rise to all the tissues of the animal and mixed up in many different ways. And, as we heard this morning, if you are clever and make your mouse mutant in a gene that is unable to make the pancreas, then what happens is those embryonic stem cells from the rat can actually take over the pancreas. There's an empty niche, and the ES cells fill it. So you can generate a mouse with a rat pancreas, and in fact you can do the reverse experiment and generate a rat with a mouse pancreas. If you think about doing that, not in rats and mice, but doing it in human and another animal, then the concept is that you could generate human-derived organs in an animal and really deal with some of the ongoing issues about organ transplants. So we talk about stem cell therapy, but none of the stem cell therapies I showed you on that list really involve organ replacement. It's a much more complex process, and yet we still know that we need organ transplants, heart transplants, and many other transplants. There aren't enough donors. So could we generate organs in an animal? And it's really the vision of these two scientists here. I always call this their vision. Hiro Nakauchi in Japan and in the US, and Juan Carlos Ipsabura Belmonte in California, share this vision of using this interspecies approach, using pigs or sheep, large animals, in which you would take their blastocysts, make the animal, using CRISPR gene editing, have a gene that knocks out the pancreas, and essentially put human stem cells, human pluripotent stem cells, into the animal blastocyst and generate a pig with a human pancreas. Well, is it possible? The first paper was published in 2017, and I will just point out that, in fact, if you read that paper, the extent of contribution of the human cells to those pigs was extremely low in any tissue. And they were not pigs that had a mutation in the pancreas, for the pancreas at this point. They were just normal pigs. So I don't think that we're anywhere near having their vision come to reality. We don't know how human pluripotent cells will behave in animal blastocysts. We think that they have to be well matched, and somehow the human and the pig at this stage are not well matched. But that doesn't mean to say that in future they might be able to better match the cells, and that you could think about making human-animal chimeras in this way. And so while it's a nice idea to generate a human organ and generate a human pancreas, although as was stated it would have pig cells mixed in it, it raises other issues because the cells you put into the blastocyst are pluripotent. And we know that pluripotent cells give rise to every cell in the organism. So you're going to generate a chimera in all tissues. If it works well, all the tissues of the pig will contain human cells. And so this really leads us to the question of how would we regulate such chimeras if they were successful? If the cells contribute to specific tissues, like extensive contribution to the brain, at what point do we consider a pig with human neurons and human cells in the brain starting to move too close to possible human cognition in an animal? What about generating human gametes in pigs? I don't have an answer to this because I'm not sure, with what we know about pluripotent cells, how you would actually regulate this approach. But this is something that is clearly on the agenda for future discussion. So that's what we can do with stem cells. But human embryo research goes beyond the generation and the use of pluripotent stem cells, whether for regeneration or for chimeras. Human embryo research itself really is a very important area of current research. And there's a lot of interest and need to study how early human embryos develop. So back to the blastocyst and perhaps beyond. Why would we want to study early human development? Well, first of all, if we can take early human embryos to the blastocyst stage and perhaps beyond that to the early development of the fetus in culture, it would give us fundamental understanding of a key stage of human development that we can't get at any other way. This is the stage when it's wrapped up inside the uterus, and indeed, patients do not even know they're pregnant. We need to understand the differences between mouse and human. I've spent all my life working on mouse embryos, and we now know that although there's a lot of similarities, there are differences. And we need to understand those differences to understand stem cells. We need to understand how to make better human stem cells and their differentiation. IVF technologies, how we began working with human embryos, still need to be improved. We need to understand the origins of germ cells, as we heard this morning. And it would be very important if we could do it to really model the early implantation process in the human in culture, because that's the stage at which early embryo loss is extremely high in human pregnancy. It's also the stage at which the placenta begins, and we know that placental insufficiency and placental problems underlie most pregnancy disorders, and we have very little understanding of their early origins. And indeed, we could get a better understanding of the origin of developmental defects in humans if we could genetically edit those human embryos and study the effect of different gene mutations on early development. So a lot of reasons to think that it would be useful to be able to go beyond the culture of human embryos to the blastocyst and culture beyond that stage. We've heard that there is a 14-day rule that's been imposed across many jurisdictions, that it's not allowed to culture a human embryo, an intact embryo, in culture beyond 14 days. And so until recently, nobody really tried that because we didn't have culture systems that allowed such development. Recently, two groups in 2016 described an improved, not a great, but an improved culture system for human blastocysts that allowed the embryos to grow across the implantation period in culture in a more or less two-dimensional manner. So this is a picture taken from one of those systems. If you look at the top, you can see some degree of organization, and then with artist license on the bottom, they have taken these pictures and told you that they have this very nice organized structures with the epiblast, the endoderm, and the beginning of the amnion. And as I say, with a little imagination, you can see that here. So they're not perfect, but they certainly are going beyond the stages that people have been able to do before. But this kind of research has actually stalled at this point in time. We're not seeing a lot more research on culturing human embryos. Number of reasons for that. Obviously, access to human embryos is an issue, especially if you're not allowed to create embryos for research. And the 14-day rule. These cultures were all ended before 14 days, and none of those cultures had actually reached the early primitive streak stage when the beginning of embryo patterning occurs, when the beginning of germ cell formation occurs. So if we can't culture human embryos, is it possible to mimic human early embryo development going back to stem cells? So can you develop self-organizing stem cell cultures that mimic events in the early embryo? And of course, it's a bit circular, because how would you know you'd mimicked it if you weren't allowed to study normal development? But we'll come back to that. So there have been a number of advances actually using pluripotent stem cells human and mouse, to generate organized structures that are giving us some information about the patterning interactions that occur in cultures and could mimic what's going on in the embryo itself. So a number of groups, a couple of different groups, have used micro-patterning cultures where embryonic stem cells are plated out on these sort of circular micro-patterns. And what you see is that these structures organize radially into something that could be considered, if changed its topology, to show the organization of the cells that develop in the primitive streak itself. So this is a nice system, it's reproducible, and you can start measuring signaling gradients and so on that can specify cell types. Other groups, this is a group in Cambridge, has shown that you can take embryonic stem cells, and a mouse in this case, in a three-dimensional structure to produce something that doesn't look really like an organized fetus that you might think about, but it has anterior, posterior, dorsoventral, and even potentially left-right patterning. So it's beginning to start to put together the cells in the right way that an embryo would develop. But none of these are just with embryonic stem cells. A more interesting one, I think, from the embryonic stem cell system is actually human, and this is work from Jianping Fu in Michigan, where he's used a microfluidic approach, a very controlled approach, to take human ES cells and put them in these little aggregates, which in the right conditions start to make a cavity and really begin to look as though they are like the amniotic sac, with epiblast and amnion developing in these hanging drop cultures. So this is, again, an ES cell-derived system, but now three-dimension and highly controlled. So this is a system that also, I think, has a lot of capacity to think about looking at embryo patterning. But what we're seeing here is all of these are with pluripotent stem cells. I want you to remember the blastocyst and realize that there's something missing from these cultures. And the thing that's missing from those cultures is that they're all only derived from the epiblast cells. They are the pluripotent cells, but if you actually wanted to mimic normal embryo development, be it the mouse or be it the human, you need three cell types. The blastocyst has three cell types. There's the outer cells, trophectoderm, which give rise to the placenta, and then both the epiblast, the pluripotent, and the primitive endoderm within the inner cell mass. ES cells come from the epiblast, and in a mouse, here we see that these are all mouse cells. They contribute to an entire fetus, but they don't make the placenta and they don't make the yolk sac. So if you wanted to mimic normal human embryo development, you're missing cell types. Well, what we know in the mouse and now in the human is that you can actually derive stem cells from the other two cell types of the blastocyst. So trophoblast stem cells can be derived from the trophectoderm. They grow indefinitely in culture. If you put them back in a mouse into the embryo, they contribute to the placenta. Zen cells come from the primitive endoderm. Put them back in a chimera, they make the yolk sac. So now we have three cell types. What happens if you mix them together? So this is now a very active area of research in the mouse. But I just want you to know that very recently, human trophoblast stem cells have been derived, and we've derived several years ago now human Zen cells. So there's the potential to what I'm going to show you now could be translated into the human. So what can you do? So Magda Zernitscher-Goetz's lab recently has taken ES cells, TS cells, and Zen cells, and with a little bit of trickery and aggregation in the right combinations, I defy you to tell you which one of these is a normal embryo and which one is actually an aggregate of ES cells, TS, and Zen. So we're seeing, I think this is the normal one, and this is the aggregate. You're seeing here are the epiblast cells, these are the ES cells, these are the TS cells, and the white ones are the Zen. And they've organized themselves into something that resembles very much an early post-implantation mouse embryo. So now you have a situation where you can make large numbers of what she calls artificial embryos. I would call embryo aggregates, to follow my predecessor. And they are really an interesting way to look at the interactions between these three cell types. But obviously in the mouse, we don't necessarily need to do this because we can study the embryo itself. What would happen if you did this in humans? Well, you should also know that although these make things that look like embryos, if you put them back into the uterus, they don't implant. Well, they implant, but they do not develop. And it's really post-implantation. It's not replicating the blastocyst. Can you make a blastocyst from mixing stem cells together? So recently, Nicolas Riverin's lab has been able to do just that, again in the mouse, taking ES cells and TS cells, aggregating the ES cells first, and then in little wells here, just dropping TS cells on top. And in these conditions, they were able to show these cells can organize into structures that look very much like normal mouse blastocysts. And you can do it in large numbers. These are the blastoids on the left, and these are blastocysts on the right. So he's able to generate these constructs. They still lack a lot of primitive endodome, and also when put back in the uterus, although they'll cause a response in the uterus, they don't develop further. But again, this shows us that the beginnings of being able to take stem cells and put them together and make embryos is not so far away. So what would happen then if we do this with human cells? I told you that those cells now exist, and those experiments will be done in the short period of time, I am sure. So are we seeing emergent properties in these stem cell aggregates that are really challenging, again, what it means to have human organismic potential? The individual stem cells alone cannot make a human organism, but now we put them together, maybe they can. So what do we do about that? This is an ongoing discussion. Again, in the US, there is some concern that NIH is going to put a ban on this kind of research, even though it would allow us to study human development in ways that we couldn't before, an absolute ban. And yet, it really one has to consider then, what should be the restrictions? Should there be restrictions on making the embryoids in vitro at all, if they have the potential ability to implant in the uterus? So should we stop anything that has trophoblast stem cells in it? Or is it sufficient to say this work is important to study development, but we must and will prohibit any reproductive use of such constructs? This applies in the same way that we heard about the gamete work this morning and potentially generating embryos from stem cells to gametes. Again, where do we put the restriction? And then finally, it was said that I would talk about gene editing, but I'm only going to do it briefly. So I think we've had many discussions in many different fora around human gene editing, but this is another game changer that we have to consider when we're considering the potential of the new technologies to treat and improve human health, but also with the ethical concerns. So CRISPR gene editing, we've heard it talked about. We know that this is a system derived from bacteria that is a very, very specific system that allows cutting and editing the DNA at very specific sites across the genome. It's very specific, very efficient, and importantly, actually very cheap, which means that this system has been democratized very rapidly and is now used in cells in culture, in plants, in animals, bacteria themselves, fungi, and of course, in humans. The efficiency means that we can imagine doing things with CRISPR that we could not have imagined only a few years ago. So gene therapy, somatic gene therapy, is being transformed right now and will be transformed in the years ahead with CRISPR gene editing. And the targets for somatic gene editing, that is either taking cells out, gene editing them and putting them back, such as skin cells or blood cells, or even gene editing in the human themselves, such as liver, lungs, the eye. We're seeing potential gene editing to cure genetic disease moving very rapidly to the clinic for two reasons: the efficiency and the availability of vectors like AAV that will take the CRISPR components and really allow them to integrate at high efficiency into the appropriate tissues. So it's an amazing time. The one that I always think, to me at the moment, is the most amazing, because when asked in the past, what do you think would be the last target that you would think would fall to gene editing or gene therapy, I always used to say Duchenne muscular dystrophy, because that is such a dispersed disease. Every muscle, including the heart muscle, in an animal or in a human or in an animal, in a human is affected by the mutation that causes the loss of dystrophin. So how are you going to fix that? And yet, the efficiency of CRISPR gives us hope that even that may be accessible. So most recent work in mice, and then more recently from Eric Olson's lab in a larger animal, a dog, has been able to show that you can take AAV and carry in the CRISPR components, and with a particular mutation where it's fairly easy to correct the mutation using CRISPR, they were able to show, first in mice and now in dogs, that if this is the mutant muscle, and this is the muscle that's been infected with the AAV virus carrying the CRISPR-Cas9, you see that dystrophin, which is the green stain here, is now on the surface again of not every cell in these muscles, but a very large number, and you do see improvement in muscle function. This is really incredible, and there's a big push and a big company push as well to move this to the clinic. So somatic gene editing is here. What about germline editing? Why would we let people have diseases if we could prevent diseases? We've seen this picture before, the designer baby. And I've put on it the concept that we would use this approach in embryos the way we and many others do in the mice. In mouse embryos, you can make genetic alterations using CRISPR at close to 100% efficiency. It's incredibly efficient. If you did that in humans, we could prevent cystic fibrosis or Huntington's disease if we had the appropriate embryos and the appropriate tools. But where do we draw the line? Is it okay to do this, but not necessary to make a sprinter? Is it okay to enhance IQ if we knew how to do that? Would it be okay to lower the risk of Alzheimer's? We do know some of the genes are involved in that, breast cancer. There are other ways that one can think of enhancing the genome. So the question is, where do you draw the line? Do we move ahead? And where do we draw the line? Many groups, many working groups, and ongoing working groups now, an international body of the academies looking at setting out guidelines. WHO has a working group on this that are really trying to answer these questions more specifically than I would say most of the working groups up to now have done. Most working groups have said, well, you know, we could do germline editing, but only when it's safe and only when we know exactly what we're doing and only when we decide as a society which modifications are appropriate. Well, okay, but how do we make those decisions and where do we draw the line? And of course, this was brought into high contrast by the recent announcement from a researcher in China and a recent announcement from a researcher in Russia that they feel they're ready to go ahead using this approach to modify the ability of cells to respond to the HIV virus. And you know that this caused a lot of consternation in China and around the world, and actually I think has done one good thing, that it has really catalyzed, again, the concept that we have to have international guidelines to really set out the road forward for germline gene editing. So here we are, the science of stem cells, embryos, germ cells, gene editing, chimeras, it's all moving at a very fast rate. It's very hard to keep up. Every day there is something new. And inevitably, just inevitably, legal, ethical, policy implications lag behind. I want to make one point, because this is the way it sits in Canada. If you have legal prohibitions, other than perhaps nuclear cloning, I think reproductive cloning, any other legal prohibition that you might put in place, you might want to revisit in the future. And every time you put legal prohibitions in place, it becomes very difficult to change them. And in Canada, we have a number of legal prohibitions which do actually impinge on some of the research areas that I just described. It's better to have a responsive regulatory framework rather than a set of clear legal prohibitions. But, as has been said many times, it's not sufficient for scientists, lawyers, ethicists to sit around and discuss these issues. We do need to engage the patients, the families, public, policymakers to debate how and when to draw the boundaries. Thank you.